Hi everybody, welcome to chapter 4. Here we're going to look at cell cycle and mitosis. We're not going to cover everything in this chapter. We'll start out with an overview just to get you back to where you should be with General Biology 1, cell cycle knowledge. And then 24.2, 24.3 will break up into several videos to look at nuclear division and cell division. And then we'll take a look at the regulation of cell cycle processes and finishing up with a short video on apoptosis. So let's get going really quickly with an overview. When we think about cell growth, that is a cell getting bigger, that's generally accompanied by cell division, where one cell gives rise to two new daughter cells. And so to do that, genetic information in the nucleus must be accurately duplicated. That's done in a process called DNA replication during S phase. We'll leave the mechanics of that to genetics. That's really part of molecular genetics. And then the DNA has to be segregated and carefully distributed into two new daughter cells. So in doing all of these processes of growing, replicating DNA, and then separating the DNA and dividing the cell goes through a series of stages called the cell cycle. And we'll focus on two of those, mitosis and cytokinesis. We'll talk a little about G1 and G2, and we won't really talk about S phase. So let's take a look at that graphically. The cell cycle really begins when we think about one cell dividing into two cells. So if we follow this cell through the cell cycle, it's going to pass through G1 where it's going to grow and get bigger. And then if we think about just one chromosome in a cell, obviously there aren't any cells with one chromosome. Let's think about prokaryotes, and that wouldn't really apply to this because it's a eukaryotic cell division. If we think about a cell with one chromosome, during S phase that gets replicated and we end up with a chromosome containing a pair of sister chromatids. And then we go through G2, and then when we enter mitosis, the two sister chromatids are separated into what will be two new daughter cells, and then cytokinesis in M phase will separate the two cells to give us two new daughter cells, and we can repeat the process to produce another two daughter cells. Important here, nuclear division or DNA segregation is the mitosis part of M phase, and the cell division part is called cytokinesis. So M phase, mitosis phase, has two stages: mitosis and cytokinesis. So don't get all those three terms confused. So let's just take a look at the process a little bit more detailed in the context of the cell cycle. Let's look at the process of M phase, I should say. So at the beginning of mitosis, chromatin kind of starts to fold and condense to produce these visible chromosomes. What we'll also see is that we have duplication of the cell's centrosomes, so that we go from having one centrosome, a pair of centrioles, to having two centrosomes, that's four centrioles. So that happens in S phase as well as DNA replication. Like I say, chromosomes have been replicated, so each chromosome has a pair of sister chromatids. As we move into mitosis, the mitotic spindle will form, and the mitotic spindle will distribute the sister chromatids in a chromosome to opposite ends of the cell. So that when the cell divides, we end up with two new daughter cells, each with exactly the same genetic complement as the cell that started the division process. And so that process occurs through metaphase, anaphase, and then we get into the end of mitosis, and then we divide the cell into two new daughter cells. So the amount of time that cells spend in this process of mitosis, or in mitosis and cytokinesis and M phase collectively, I guess, is called the Mitotic Index. And that can be determined just by looking at a group of cells and determining the proportion that are in mitosis. And you can do this visually down a good light microscope. You can actually see the cells that are in mitosis because of their condensed chromosomes and because of structures like the metaphase plate, and because of cells that are going through anaphase tend to be elongated. You can see the chromosomes separating. So in cultured mammalian cells, that's about 3 to 5% of the cell population, maybe as high as 10 with certain cell types. And so that means that cell cycle time is probably anywhere from 30 minutes to an hour, I would say. So anywhere from 30 minutes to an hour, depends on the cell type. Sometimes it's quicker, sometimes it's less. 30 to 60 minutes, I would say, on average. So that means that most of the cell cycle, most of the time a cell spends in the cell cycle is in G1, S, and G2. So G1 is quite variable and depends on the cell type. S phase obviously depends on the amount of DNA to be replicated, but that's pretty consistent, taking up just over a quarter, maybe around a third of the cell cycle time. And G2 is usually much shorter and less variable. So collectively, the whole process takes up about 18 to 24 hours, and so it depends on the cell type, obviously. Now, probably the most important thing in G1 is a decision the cell makes about whether it should divide or not divide. And so cells can wait in a stage called G0, which would be kind of like represented by a little cycle out here to the side. Cells can wait in G0, not entering the cell cycle, for appropriate growth signals so that they can enter the cell cycle. So we'll look at some of those growth signals and their role in driving cells into G1 through SG2 and then into M phase. So cells, other cells which leave the cell cycle and exist in this G0 state, they will terminally differentiate. So most of the cells in your body once you're an adult are terminally differentiated. The cells that are in the cell cycle tend to be in stem cell populations, although there are other cells outside those populations which do also divide. So if we look at the process of mitosis in a little bit more detail, we can correlate the diagrams at the bottom here to what's going on in actual cells being visualized by confocal fluorescence microscopy. So interphase, the chromosomes are decondensed, and we replicated our two centrosomes in S phase. As we go into prophase, the chromosomes begin to condense, and you can see them becoming a little bit more easily observed here in the microscopy image. You can just about make out two centrosomes here. Microtubules are in green, and you can just about make out the two yellow dots here, which are the centrosomes. And so the centrosomes are beginning to separate, and the mitotic spindle is beginning to form. The nuclear envelope will also start to break down at this point. As we go through pro-metaphase, the nuclear envelope is completely disintegrated. The chromosomes have fully condensed at this point, and the chromosomes are attaching to the mitotic spindle, which is nearly completely mature at this point. As we go into metaphase, the attached chromosomes have aligned up on the metaphase plate in the middle of the cell. The mitotic spindle is completely formed, and in the next step of anaphase, you can see that the chromosomes, the sister chromatids in the chromosomes, are being pulled towards opposite ends of the cell, and so the mitotic spindle is pulling the chromosomes, the sister chromatids, to opposite ends, and the cell is also all elongating. So we'll talk about those processes when we look at anaphase in more detail. And then we go into telophase, where a structure called the cleavage furrow forms, and that will result in a pinching of the cell into two new daughter cells using the non-muscle myosin 2. And then the chromosomes will begin to decondense and go back to their more stringy kind of appearance, and the nuclear envelope will reform. So that's a very quick overview of where you should be at in terms of cell cycle knowledge coming out of General Biology 1. I'll wrap it up there, and I'll see you next time when we can start looking at some details of nuclear division as well as cytokinesis. I'll speak to you soon. Bye for now.